Okay, so recording in progress. So that seems like a very good moment to start. So welcome everybody. Thank you for being here at this moment. And it's a really exciting time of the year where we have the new members of the BRI being given a chance to tell us about what they are doing. And I hope that this serves to know each other a little bit better and for all of us to understand how the BRI is growing and how the neuroscience community at UCLA is growing. So we're very excited today to have 3 short talks given by Idan, Aparna Bhaduri, and David Clewett, who will all take about 15 or so minutes to tell us about what their research is. And I didn't ask back, but I assume because the talks are relatively short, that it might be better to take the questions at the end rather than during the talk. Okay, I see some nodding. So I'm going to just give a brief introduction to everybody before their talk. And so let's start with Idan, who's at the very top and I understand has to go and teach afterwards. So without much further ado, Idan comes to us from Israel. He got his master's degree at Tel Aviv University and then came to the United States to get both his PhD and do postdoctoral work at MIT in the lab of Evelyn Fedorenko, where he started a lot of the work that he's going to tell us today about. Idan joined UCLA in, I think it was 2019 for the Department of Psychology. And now since about a year, he has a joint appointment at the Department of Linguistics. And that's really relevant. You will see from his title, but also from the talk he's going to give to us today. So without further ado, Idan.
Thank you. And thank you all for coming.
Who is Aparna Bhaduri, and Aparna went to Rice University to get a BA in political science, then decided actually at the same time to also get a BS in biochemistry and cell biology. She then moved for her PhD to Stanford University to get a PhD in cancer biology. And finally, before she came to UCLA, she was in Arnold Kriegstein's lab at UCSF and really sort of started the work that she's going to talk to us about today. She's been at UCLA officially since January of this year. So she really sort of started with us at the end of the lockdown, and hopefully now that we're reopening, you can finally really start your lab for real. So thank you, Aparna.
Thank you so much for that nice introduction. I appreciate it. And I will just share my screen here. Thank you everyone for being here, and really a wonderful first talk. I learned a lot about language processing and how the experiments are done to do that. I work in a very different area of neuroscience, specifically developmental neuroscience. And the focus of my lab is understanding how neural stem cell specification occurs. And specifically, I am fascinated by the ability of a neural stem cell to give rise to the huge diversity of cell types that we see in the adult human brain. And I'm interested in understanding how this happens across areas and regions and time points of development, and also how we can use in vitro models of the developing human brain, which is quite distinct from those of other model organisms. So using systems like cortical organoids to do some of this work, and then also exploring a very alternative situation where neural stem cells are reactivated in a cancer like glioblastoma. And so a lot of my work uses single cell RNA sequencing, and we're hoping to expand this to other modalities, which we can look at epigenetics and metabolomics, both in bulk and at the single cell level, and really answer some questions about what are the cues that result in fate specification of this neural stem cell. So I'll present two small vignettes of one project at the end of my postdoc that I worked on to characterize some of the areas using single cell RNA sequencing of the developing human brain. And a second talking about slightly older work, but that is foundational for the next stages of my lab, looking at the reactivation of the stem cell in glioblastoma. So the project that we sought to do to look at regions of the developing human brain was funded by the Brain Initiative. And we wanted to be able to sample different areas of the brain as well as different areas of the cortex during developmental time points. And for the interest of time for today's session, I will talk just about the neocortex, where many of my interests lie. So in the neocortex, we were able to sample approximately 6 different cortical regions. And from this, we were able to identify 400,000 different cells. Now, this is the overall distribution of the numbers of cells that we were able to retrieve. And because I know this is a diverse audience, I'll briefly explain these UMAP plots, which will emerge across the talk. So what we do when we have single cell sequencing data is we take each individual cell and we have the number of each gene counts that we can find. And so we basically have a matrix of counts with each cell and each gene that is detected in the analysis. And from there, we're able to use a couple of computational strategies to reduce the complexity of the data. And ultimately, we generate clusters. And we can do this using different parameters, so either looking at clusters unbiasedly or looking at differences between areas. But we ultimately visualize this in, sorry about that, but just in 2 dimensions. And so this is called a UMAP plot. And this is the most reduced form of the data, where we're looking at the similarity between individual data points in these UMAP coordinates, and each dot is a point. And what we can see from the whole data set is that we have various individuals, with some individuals being enriched at earlier or later time points, but also that there is mixture between most individuals, and we can see some segregation by brain region. So as I mentioned, we'll focus in on the neocortex. And when we look at this data set, we see that we identify known and expected populations. So we're finding radial glia and neurons and dividing cells and interneurons, et cetera. And one of the first questions that we had was, how are things changing across the cortex? So as a little bit of background, the process of mouse patterning and human patterning is relatively similar, with many of the core transcription factors that respond to early morphogenetic events that set up these gradients. But as you can see, even from these 4 transcription factors, there are some small differences between some of the expression patterns. And these initial events of patterning set up these transcription factors. And then we have the cascade of normal cortical development, where radial glia give rise to intermediate progenitor cells, and then give rise to the layers of the cortical plate, which mature and become the layers of the cortex. And one of the long-standing questions in cortical development is, once you have these initial patterning molecules, FGF8 is one of the most famous ones, but there are others as well that set up these anterior to posterior gradients, as well as medial to lateral gradients. Are the radial glia that are giving rise to the rest of the cells predetermined in their identity? Do we know what they will become? Or are they dependent on some of the extrinsic mechanisms that are coming from sensory input, including from the thalamus? And this is a question that we do believe that there's probably some in-between explanation, but we wanted to see how our molecular data and signatures at each of these different time points would be able to inform us about what's happening and how it's happening. So when we look at each of these cell types, and you can see the clusters that belong to each of these major cell types, we had a total of 138 clusters, and you color them by the area from which the sample was dissected, you can see that for the most part, there is actually intermixing across all of the clusters, but there are a few clusters that are strongly enriched for either PSC in red or V1 in blue in this particular diagram. And so one of the things that we asked is, overall, how are we seeing this cascade happening? And we focused in on the excitatory lineage, so the radial glia, the IPCs, the neurons, but then also some of the early glial populations, such as OPCs. And in this plot, each of the dots is a cluster, and it's localized to that original UMAP space, but it looks different because it's the average of all of those UMAP coordinates. And what we were able to see when we quantified the relationships is that there's a lot of strong relationships between the same cell type. So this suggests that our clusters are one way of designating the cells, but there's probably some intermixing between these clusters. And then also that there is strong correspondence between the intermediate progenitor cells and the neuronal populations, which was interesting because it's typically been thought that these IPCs are maybe more like the radial glia. And what we are seeing is that the radial glia are strongly activating the radial glia signature, whereas neurons are more defined by a strong deregulation of this progenitor signature, perhaps because maturation is not yet complete for these cell types. We validated that some of the known transcription factors, some of which I showed before, are in fact expressing the pattern that we would expect. And then we wanted to know what is happening across areas. And so again, you see a constellation plot here. This time each dot is colored by cell type and is annotated by the area it's from. And what started becoming very clear here is that the connections between these different areas are pretty intermixed, but there's strong mutual exclusivity from prefrontal cortex and V1 already in the radial glia, and that the relationships between the intermediate progenitors and the neurons cascades out as this moves along the differentiation axis, suggesting that you have a very strong initial intrinsic specification of frontal versus occipital, but perhaps in between things become more defined later on. We wanted to know how this is changing in terms of the numbers of genes and the specificity, as well as how it's changing over time. And what we see is that although there are a lot of genes that are differentially expressed in the radial glia, they are much more specific in the neurons and the intermediate progenitor. So this fits with that previous model, but that they're highly dynamic. So the genes that are PFC specific in radial glia, some of those will be preserved in IPCs, but many of them will not. And this was one of the surprising findings of this work, is how dynamic these signatures are, not only across cell types, but also across stages of development. From this, we were able to identify a few transcription factors, and these are of keen interest to study moving forward, to think about how this may be regulating aspects of the developmental sequence and that dynamicism that we're seeing in specifying area. We also sought to do spatial transcriptomics to validate some of these observations, and we were able to collaborate with a company called Rebus in the Bay Area, where we were able to look at 31 genes simultaneously across different samples of the developing human brain. And so here we show a section of somatosensory, but we were able to look at 4 cortical regions. And this was really exciting, and we feel that the data is high quality, but it's also very confusing. We are able to validate some previous observations. So we've previously seen that BCL11B and SAPB2 are more co-localized in frontal regions, but more mutually exclusive in occipital regions. But when we look at some of the new patterns, we see that while they do validate some of the expected distributions, there's also a lot of intermixing and changes in laminar identity. And this, paired with some of the co-expression networks that I'll show you on the next page, I think are an area of great interest for moving forward in terms of what does spatial identity mean and how can we potentially model this in cortical organoids. And so as you can see here, there's a lot of differences in the level of interconnectedness that you're seeing. And these are questions that are raised by the spatial data in addition to validating some of our previous observations. So just to close up this part of the story, we see that there's strong signatures that we can identify from the single cell related to differentiation, maturation, and spatial position. But there's also now a ground truth data set that can let us follow up on some of our open questions, such as how, when, and by what mechanisms is the specification enabled? And we can follow up on some of the targets that we found from previous work. And then how could this be manipulated? I highlighted some of the transcription factors that are preserved between human and mouse, but there's also a lot of differences when we compare our data sets to others that have been performed in mouse, and how can we do this in a cortical organoid? And then also thinking about ways in which we can improve that system to better model some of the maturation and transcriptional reprogramming that we see over time as a human brain develops. So these are areas that we are pursuing now in the lab. And I'll just take a small amount of time to highlight some of our thoughts on glioblastoma. So as some of you may know, glioblastoma is the most aggressive brain tumor. And it's this aggressive and problematic because of its high rate of recurrence, which is often mediated by the recurrence of tumors at sites very distant from the original tumor. Now, we are definitely not the first to be interested in the molecular underpinnings of this cancer. And one of the first cancer genome atlas studies looked at bulk RNA sequencing of hundreds of tumors and identified 4 categories. But unfortunately, these 4 categories were not predictive of clinical outcomes. And one of the first single cell studies showed why. Each individual tumor at the single cell level was creating a mix of these different identities. And more recently, people have expanded the single cell approaches that exist and have seen that there can also be transitions between states that are identified at these time points. So this is kind of where we came in and where we wanted to add in our own single cell RNA sequencing, but use it referencing some of these atlases of normal development, as well as a few data sets from the adult human brain that we had. And from these data sets, we were able to annotate the tumor bulk with some populations that were expected, as well as some that were a little bit strange, but maybe specific to tumor populations, including those that are mixing progenitor and neuronal identities. What I really wondered was, could we annotate the cell type of the cancer stem cells? People have annotated markers that can be purified, that can then give rise to tumors in xenograft models. We wondered, could we identify with our new knowledge of developmental cell types, some of the cell types that associate with this? However, the theme of heterogeneity continues, and we were able to see that there were many different cell types that were represented in these tumors. But we were intrigued that one of these cell types was a radioglia type. And as I mentioned, our lab was interested in studying these progenitor populations. And when we did some comparisons to subtypes of radioglia, we saw that they most resembled this outer radioglia cell subtype. Now, outer radioglia are interesting because they undergo this jump and divide behavior. And we wondered if we would be able to see that in tumors. So these are images from actual glioblastoma tumors. And indeed, we were able to find a correspondence to this outer radioglia behavior, including the production of cells that gave rise to other cells, so daughter cells that were proliferative, which you can see in the second video on the right. And this suggested to us that there are multiple cell types that probably are associated with this progenitor identity. And this outer radioglia type may be one interesting type. So using that PTPRZ1 marker, which was central to its network, we isolated these from patient tumors and transplanted them into cortical organoids. And this is an approach that I'm really excited about, and we're trying with a lot of collaborators here at UCLA, because it's a way to grow these in vitro in a way that seems to maintain some of their heterogeneity. And what we observed was that the outer radioglia-like cells were capable of giving rise to cell types that didn't exist in the original sort, but consistent with the heterogeneity, so were the negative populations as well as the unsorted populations. Now, PTPRZ1 has been shown to signal through this ROROC pathway, and this was interesting for us because that was the same pathway that has been implicated in mitotic somal translocation, or that jump and divide behavior that I showed you previously. So we did knock down PTPRZ1 and saw that there was a reduction in the MST, or the jump and divide behavior. And this was interesting to us because it also correlated to a decrease in invasion. So I know I flew through some of this data pretty quickly, but it is published, and I think it sets the stage for some of the things that we want to do moving forward, where we're really interested by this heterogeneity and are seeking to integrate all of the published data sets that exist and are identifying really granular heterogeneity with better annotations compared to what we've seen before, what we've been able to do. And this is an integration of 67 tumors. And this is what we're kind of following up now in the lab to see if we can not only mechanistically further characterize PTPRZ1 as a potential therapeutic target, but also use some of these strategies to better understand the cell of origin as well as the lineage relationships between these populations. So with that, I would like to thank the people in Arnold Kriegstein's lab who were very integral to much of this work, as well as the people in my new lab who have come through, and it's been wonderful to get to know people in this community. So with that, I'd be happy to take some questions.
Thank you very much, Aparna. That was awesome and really nice to see that you're recombining both your PhD and your postdoctoral work, so both looking at development and cancer. So that's really nice. Questions? just unmute yourself or raise your hand, and then we're going to call on you. Dr. Gupta, let's see whether you got sound this time.
Yeah, am I audible? Yeah. Good evening. So my question is, how does your study translate into the actual, like at present we have multimodalities treatment for glioblastoma, like tumor decompression and immunotherapy, bevacizumab. So how does your therapy will help us further in this glioblastoma patients? Like when you do a molecular subtyping of these patients. At present, we are doing the P53 and the TRT mutations and working on all that. How does your study will lead us further?
Yeah, thank you so much for the question. And I think that that's a really important implication of any research into tumor biology. I don't think we're yet at the point where we can leverage the subtype heterogeneity for clinical outcomes, but my hope is that by using some of the systems that we have here now, we can really understand the lineage relationships between each individual population and how it relates to recurrent tumors. And that the dream would be that one day you would be able to use single cell RNA sequencing to identify which populations in a tumor would be problematic and then specifically target those therapeutically. And I'm hoping that some of our organoid model culture systems can be useful for some of that drug testing as well, where we could do it on human cells and actual patient cells, and see what types of approaches could be directly targeting the tumor.
Any more questions? Otherwise, if you have questions for the speakers, I'm sure they will be happy to answer more questions via email, collaborative suggestions and things like that. So we get to the third speaker of today, who is David Clewett, who did his BS in biopsychology a little bit north of here at the University of California, Santa Barbara, then moved very close to here for his graduate studies at this other university closer to downtown with Mara Mather, and then decided to have a stint on the East Coast, went to New York University to do his postdoc with Lila Davachi, and then came at last year and joined the Department of Psychology, where he's now an assistant professor. David, and his title is Arousal Mechanisms Shape the Selectivity and Structure of Memory.
Awesome, thanks so much, Felix. So hi everyone, my name's David Clewett, and I'm an assistant professor of psychology in the cognitive area at UCLA, as Felix mentioned. And I'd like to begin really quickly by thanking BRI for this amazing opportunity to join this institute, and for giving me the chance to work alongside all these members whose work I really admire. It's a true privilege and honor to be here, so thank you. So like all science, I'm going to begin today with some observations about the human experience. So in everyday life, we're constantly bombarded by new sensory information. However, because our attentional and memory resources are limited, we cannot process and hope to remember everything that we encounter. So this raises two fundamental questions. First, how do we select which information gets committed to long-term memory? And second, how do we organize all of these disparate elements of experience into these meaningful memory representations? And so my lab's research is built upon the physiological construct that arousal is going to facilitate attention and memory. And for decades, these effects were primarily studied using individual items, such as an image of a snake or the sound of a scream. But this focus on individual items led to three critical blind spots in the learning and memory literature. First, we know that arousal does more than simply enhance emotional representations. Rather, it can spill over in both space and in time to enhance or impair how we process neighboring neutral information or details. Second, most research has focused on how arousal influences how we acquire or encode new memories. And by comparison, relatively less attention has been paid to how arousal can selectively shape the consolidation of more distantly encountered events. And third, of course, arousal is happening all the time and not just in response to emotional or distressing events. Rather, our arousal levels fluctuate continuously throughout the day, suggesting that they might be able to translate the structure of our experiences into these meaningful memory representations. And so in my cognitive neuroscience lab at UCLA, we seek to understand how emotional and everyday arousal influence what we remember, so the details that we selectively attend to and are able to remember later on, how we remember, so the ways in which our continuous experiences become transformed to these discrete and memorable episodes, and finally, when we remember, the dynamic arousal and brain states that are induced before, during, or after a motivationally significant event. And in my lab, we're particularly interested in neuromodulation, so the role of different catecholamines like the dopaminergic and noradrenergic systems in modulating memory processing in both the amygdala and the hippocampus. And today, I wish I could describe a lot more of our data in the lab, but I'm going to showcase two individual studies that I think capture or represent a lot of the research themes in the lab, as well as highlight some of our more recent work. So as we all know, emotional events, of course, tend to form our most vivid and enduring memories. So for example, imagine that you're hiking in the woods when suddenly a bear appears and threatens to attack, which would be very unfortunate. Well, decades of research suggest that a surge in arousal and the release of stress hormones is going to help transform this experience into a lasting memory. As such, you'll be able to learn that bears are a threat, and this will help you avoid similar dangers in the future. However, the effects of emotion on memory are much more complex than that, and they go beyond the simple enhancement of threatening representation, such as the bear itself. Emotional arousal can actually spill over to also influence the retention of details that may seem trivial or seemingly irrelevant at the time they're first encountered. So to put this idea in context, imagine that earlier in your hike, you were walking along a river and you encountered several things. You may have noticed these strange markings along the path or an old camping lantern laying in the grass. Well, because neither of these things were particularly salient, they'd likely undergo the same rate of forgetting or decay over time. But what would happen if you were to encounter something emotionally arousing or stressful shortly thereafter? Well, recent work reveals that emotional learning can selectively and retroactively enhance memory for conceptually related information. So that is, because these paw prints are conceptually related to the bear that's about to appear, that information is going to selectively benefit and receive a boost in memory compared to more irrelevant details like the camping lantern. So what we think this mechanism does is it's adaptively holding on to memories that may acquire some sort of significance in the future. And so motivated by these cool findings, we wanted to uncover the different brain mechanisms that support this kind of selectivity. And so one possibility we explored is that emotional events lead to biases in hippocampal processes that are engaged after learning. So the hippocampus, of course, is a core memory region of the brain that supports the encoding and consolidation of new episodes. And increasing research suggests that post-encoding hippocampal processes, including local neural replay of recent events or functional connectivity with other brain regions, may be biased towards preferentially processing salient over less salient information. So for instance, in one fMRI study, they showed that following associative reward learning, the hippocampus became more functionally coupled with brain regions representing the rewarded versus the unrewarded information. And importantly, this bias in functional connectivity predicted the memory advantage for the rewarded information over the less salient information. And so based on this, we predicted that these offline hippocampal processes may also privilege earlier memories that also relate to the subsequent stressor. So to test this hypothesis, we performed an fMRI experiment in which subjects performed a two-phase incidental encoding task. So in phase one, participants viewed a series of neutral animal and tool images and simply had to indicate the category of the image, so judge whether it was an animal or tool. So this is a very weak form of learning. They're just incidentally encoding these neutral items. Scenes were also interspersed, but I don't have time to go into that today. So if you're interested in talking about why, I can talk to you about that later. So approximately 6 minutes later, participants then viewed a new set of animal and tool images, except this time they were novel exemplars from those categories. And importantly, in this phase, one of these categories co-terminated with shock on 2/3 of the trials, thereby making that category emotionally salient. So this is a Pavlovian fear conditioning procedure. To assess how this affected long-term memory, we then tested item recognition for all of the items from both phases 24 hours later. And what we hypothesized was based on the behavioral study that showed that pairing one category with shock would lead to a selective and retroactive memory benefit for items from that same category. So for example, if you had been shocked on animals in phase 2 of the experiment, you'd be more likely to remember animals compared to tools from phase 1 of the experiment. And so to examine how emotional learning influences offline consolidation, we also collected resting state scans between each phase of the task. And this enabled us to basically see and assess whether post-learning changes in hippocampal functional connectivity with other regions of the brain were related to that memory advantage for CS plus category items from phase 1 of the task. And so for this, Idan described a similar approach earlier. We used a resting state functional connectivity analysis. So in this approach, we first compute pairwise correlations between the time course of activity in 2 regions of interest. And then in this case, we wanted to examine functional coupling between the hippocampus and category selective cortex. So that is different regions of the ventral visual stream that are specialized to process animal versus tool information. And so to determine how emotional learning influences how these regions communicate, we then subtracted the correlation value for pre-learning rest from the correlation value for post-learning rest. So what this subtraction does is it basically gives us a metric of how communication between the 2 regions changes as a function of the recent emotional experience. And so importantly, these functional connectivity measures were then correlated with our behavioral metric of the emotional memory effect. So how much participants showed a memory advantage for the CS plus versus CS minus category items. And so based on the previous work, we hypothesized that hippocampal connectivity would be biased towards the cortical area that was representing the CS plus or salient information. So for instance, if animals were shocked in phase 2, we would expect those participants to show greater coupling between the hippocampus and CS plus area versus the area representing tools. And indeed, that's what we found. So the more people showed this selective and retroactive memory bias for emotional items, the more likely they were to show a shift towards communication between the hippocampus and CS plus cortex. And this effect also was not observed for CS minus cortex. So together, what these findings suggest to us is that the hippocampus is help preserving seemingly irrelevant memories by enhancing its coupling with regions representing that information. And we think this might serve as an adaptive mechanism for holding on to memories that acquire new significance in the future. And of relevance to this audience, we think this mechanism is analogous in some ways to behavioral tagging models in rodents. I don't have time to go through that today, but again, if you're interested in talking about that, feel free to contact me after the talk. So building on these exciting findings, our lab is now conducting several studies to see if we can use similar arousal techniques to boost memory for different types of learning. So in the previous study, we showed that hippocampal reactivation may benefit item recognition for items that are conceptually related to a stressor. However, this specific retroactive effect has only been shown for individual items in the literature, so it's less clear if these emotional events can also retroactively increase the temporal links or associations between a sequence of items. And really, this is the true goal of an adaptive memory system, right? In order to guide adaptive behavior, what we'd really want to do is establish causal relationships between neutral things that lead up to an emotional outcome. So in this current fMRI study, we're examining hippocampal activity that occurs at emotional moments, and then how that relates to the reactivation or replay of recent sequences of information. In a separate but related study, we're also interested in examining whether we can boost recent memories using a non-emotional source of arousal. So a limitation of the recent study, of course, is that we can't go around shocking people to improve their memory. That's not only highly unethical, but not very practical. So to avoid these issues, we designed an experiment that swaps out the Pavlovian conditioning procedure in phase 2 with a mentally demanding task. And so here we're instead inducing an increase in arousal through mental effort, which is driven by the subjects having to juggle one category of information in mind during an end-back task. So if this works, this would be cool because it would provide evidence of a safe and cost-effective way of enhancing recent learning, not only in educational settings, but also in everyday life. So to introduce our other major research theme, it's perhaps most helpful to bring up something that's been on all our minds recently. And that's where the heck has our sense of time gone recently? Why has it become completely distorted during the pandemic? So as we've all experienced, our memories for the past year have become very muddied and jumbled. It's hard to remember anything, right? And there's also a lot of variability in how people perceive and remember the passage of time. So for example, when we reflect back on 2020, it might seem like the month of March had lasted forever. It just dragged on and on. Whereas by contrast, the month of April may have seemed to have flown by. So what's interesting is that these changes in time and memory aren't independent of each other. And in fact, the conditions under which time seems to contract versus expand is actually telling us something extremely important about how human memory works. And that's that the structure of our experiences or context may be necessary for remembering. So from moment to moment, our lives are unfolding like this continuous autobiographical narrative. Yet when we reflect on the past, our memories don't strictly mirror this continuous stream of information. Rather, more like chapters in a book, we usually remember past experiences as being more discrete and episodic, so broken down into these individualized memories or events. But this begs a critical question. What actually creates a new episode in episodic memory? Why is episodic memory episodic? And so in recent years, it's been shown that the stability of an unfolding context helps to bind or glue information together into a coherent event representation. So for example, imagine you're taking a stroll through Washington Square Park. Well, as you navigate through this space, there's different stable features in the environment across time, such as the green of the trees or the fact that you're in the same space, that are going to help construct this coherent mental representation that you're in the park. However, when the context suddenly changes, such as stepping into a city street, it's thought that a theoretical event boundary is going to disrupt that ongoing integration process, instead separating temporally adjacent experiences into more discrete episodes. And interestingly, one way we can query these effects in the laboratory, so the effects of boundaries, is by looking at how these boundaries affect different temporal aspects of memory. So for instance, past work from our lab shows that people are worse at remembering the order of experiences that had occurred on either side of an event boundary compared to information that were encountered within a shared event. So for example, within the park. Likewise, boundaries have also been shown to elicit this really interesting subjective time dilation effect in which experiences occurring on either side of a boundary are actually remembered as happening further apart in time than experiences that were encountered within the shared context, again, within a park. And so the way we think about this is it's basically the brain's attempt to unlink continuous experience and then push those adjacent representations into more separate episodic memories. And so while we have mounting behavioral evidence for this, the brain mechanisms that facilitate this sort of segmentation process are still kind of unclear. And so in studying this phenomenon, I began to notice that there's some interesting similarities between event cognition and how emotion affects memory. And so this inspired us to examine the potential role of arousal and arousal-related neuromodulators like dopamine and norepinephrine in facilitating event segmentation. So like other forms of contextual information, such as space, our internal arousal and emotional states may also provide a type of scaffolding for integrating information to a common memory representation. Further, we know that arousal levels fluctuate continuously throughout the course of the day and also respond to salient changes in the world. So we thought this would be a promising candidate mechanism for how we organize episodes in episodic memory. And so to test these predictions, we performed several behavioral and eye tracking experiments using eye tracking to measure pupil dilation, an endogenous marker of arousal. And here participants were basically encoding sequences of objects. And in this manipulation, we inserted different salient tones intermittently throughout the sequences in order to create these event boundaries that would subdivide the continuous sequence into these discrete auditory sub-events. So we're using auditory tones to basically create this sense of structure. And so after studying each list, we showed participants different pairs of items from the sequences they had recently studied, and we queried two different aspects of temporal memory. The first is we asked subjects to rate the temporal distance between the two items, so how far apart they felt from the previous sequence. And then also the temporal order, so they're making an order discrimination about which one had come first. And so what's interesting about these effects, I should mention, is that unbeknownst to subjects, these pairs of items were drawn from two conditions. One was a same context pair in which there was no intervening event boundary or tone switch, and the other was a boundary pair so we could examine the consequence of a tone shift on how they remember different aspects of time. And what's really important is that these item pairs had actually come from items that were the exact same distance apart. So any change we see in these temporal distance rating reflect a subjective distortion in time. So any differences in whether they felt like they were closer or farther is completely made up in the brain. And so consistent with our main prediction, we saw that these tone switches were salient and elicited a robust increase in arousal as indexed by pupil dilation. Unfortunately, I don't have enough time to go into this in detail, but we also used a principal component analysis to decompose this boundary evoked pupil dilation into these different temporal characteristics that have been linked not only to different autonomic regulatory pathways that regulate pupil size, but also different mental processes such as cognitive control versus anticipation. And we were really excited because what we found is these boundary evoked responses and these different temporal characteristics of pupil dilation also predicted both the impaired order memory effect as well as the subjective time dilation effect. So what was really cool is when we zoomed out from the local effective boundaries on these kind of phasic spikes in arousal, we also found that the temporal stability of overall arousing states throughout the course of encoding also were related to how memories were integrated. And basically what we found is the more variability we saw in pupil fluctuations between the time they encoded those pairs of items, the more likely they were to show separation as well. So impaired order memory and more dilated estimates of subjective time. And so in an ongoing fMRI experiment, what I was most interested in is seeing whether these arousal signals indexed by pupil dilation are actually reflecting underlying neuromodulatory activity in the noradrenergic and dopaminergic systems. So I'm running out of time, but this data is hot off the press and we're very excited about it. But basically what we're finding is when we look at univariate responses in the VTA-SN, so dopaminergic system, so univariate just meaning a mean increase in activity at these boundaries, what we basically find first is that the dopaminergic response correlates with pupil dilation. So there seems to be an arousal evoked response in the dopamine system. But what was especially exciting is the more that individuals showed the spike in dopaminergic activity at these salient event boundaries or context shifts, the more dilated their temporal estimates were for the distance between the items spanning those boundaries. And this was true not only at the trial level, but also across participants. So those who showed more of this effect were also more likely to show more dopaminergic activity. So this is really exciting because it supports an intuition we've all had about how dopamine may be affecting temporal memory. And that's that context shifts, boundaries, novelty, and excitement are what give rise to more memorable experiences. So right now we've been stuck indoors during the pandemic. We've been in the same context for so long working from home. So really the best way to promote memory is to get outside, experience novelty, and create those event boundaries. Creating structure is going to help you remember the past. And so sorry, I might be running a little bit over. I have one more slide. Should I just stop or? I don't want to hold people hostage.
I think just go ahead.
Just go through it. Okay.
People might leave, but some might stay. Okay,
great. So now turning to future directions, one of the main questions we're interested in is how memories may be segmented for emotional experiences. So we're basically circling back to the topic of emotional memory. And so insofar as arousal is going to function as a strong event boundary, we would expect that emotional stimuli, which tend to be the most arousing and stressful, would amplify these segmentation processes. And so in recent data in our lab, indeed, this seems to be the case. So these temporally dynamic arousal states evoked by something emotional seem to make emotional memory hyper-episodic. They're not only increasing the amount of segmentation that occurs when an emotional stimulus is presented, but those effects also carry forward in time to enhance binding within the ensuing event. So it's not only segmenting, but it's integrating and mushing the following event together into something memorable. The problem, though, is these results don't necessarily speak to the memorability of those experiences. We're showing evidence of how those memories are first structured through that impaired order memory effect. But is this actually good for memory? Is it quantitatively enhancing our ability to recall those individual items from those sequences? So that's one thing we're currently exploring in the lab. And then in a related line of work that I'm especially excited about, we're also examining how this emotional event segmentation could influence fear generalization and intrusive memories, which are two hallmark features of post-traumatic stress disorder. So for instance, based on this other research, if arousal is going to form these hyper-episodic memories, does this sort of compartmentalization by these event boundaries prevent fear from spilling over and generalizing to similar events? And so currently we're predicting that event segmentation processes may be impaired in PTSD in ways that prevent these individuals from limiting the fear to the original episode or traumatic context. So that's something we're currently exploring using pupilometry and fMRI as well. And so really quickly, I just wanted to thank the people actually doing the work, my lab, the ACME lab, Mason McClay, Ringo Huang, Jamie Greer, Camille Laxman, and Zuyan Chen. Thank you so much for your attention.